All right, please sit down. Let's start. Now, next speaker is Peter Baas.
Imagine something I think that's really important for all of us as biomedical researchers, and that's the link between basic science and disease and injury. When I started in biomedical research, it was 20, 25 years ago. My commitment at that time, like many investigators you've heard of, was really in the area of basic science. But my function from the beginning has been primarily from NIH. Now, NIH in the United States is the National Institutes of Health. And if you look at the mission of the NIH, the mission is the healthcare, the development of treatments and cures, and elucidating underlying etiology of diseases that affect the human population. So that being the case, why is it, do you think, that over these past decades, NIH has given billions upon billions of taxpayer dollars toward understanding things like centrosome implication and issues that may seem very separate from human health? Well, I recall the old days or decades past, when I was writing on the PhD, I'd like to insert a few sentences I dig in the literature and try to find reference to some microtubular abnormalities that I can make of that understanding microtubules and neurons can somehow help people in certain relations. But really, the genius of the approach has come to fruition. And really, the genius was that before we can really understand human disease, before we can really understand how to treat it, or cure it, hopefully someday, we're going to have to understand those underlying mechanisms. How do dendrites grow? How do axons act? How does axonal transport work at the mechanistic level that we've heard from these past few days? Once we understand, and without understanding those underlying mechanisms, then we'll have severe disability to really craft, to really brainstorm the kinds of therapies which will help patients. We can take the approach of the ancients and go through thousands of potential therapies of different sorts of extracts and plants and try to, by the process of elimination, figure out what might possibly help patients. But alternatively, if we can elucidate those mechanisms, then we can really craft strategies. So even for those of us who came into this as basic scientists, I think this basic science really got a crucial linchpin in ultimately solving those treating these diseases. I moved about 12 years ago. I started out at the University of Wisconsin, and I was was there for 10 years. Pretty much all those 10 years I was based in some units. There's all the time that I moved to Axons 12 years ago. I think at that time, at least personally, I could begin to detect change going on. I began to submit to some various foundations of vision at our age. Because it's not allowed what you have to in order to keep your life going, you have to seek financial support from various agencies. So there were agencies such as the Alzheimer's Association, and there was the Christmas Review Association, which supports spinal research. And I began to think creatively about how my research over the past decade could possibly help people, say, with spinal cord injury or Alzheimer's disease or other sorts of degenerative diseases. I also began to appreciate, looking at the literature, how now, after decades of work in laboratories all across the world, how now we can understand at a level we never could before how some of these diseases actually work, and really how many of these diseases, particularly the basic nervous system, really have their heart underlying mechanism with these microtubules that have been studied all these years. Lysosophaly, for example, is a birth defect that occurs with failure of the brain to appropriately laminate, to appropriately layers of the cortex. Lysosophaly is caused by mutations to lysopora, which is a protein that regulates microtubules. As we've heard about in Alzheimer's disease and other so-called neuropathies, the microtubule-associated protein tau goes around, and this can cause problems. I'm going to talk in just a very few minutes on a disease called hereditary spastic paraplegia. The chief cause of hereditary spastic paraplegia is mutations to spastic, and that is over a long time. Before it was discovered that spastic is a microtubule protein. So what I'm going to be talking about for the next hour or so is microtubule proteins. And the previous talks that I've given to the school and the workshop, I've introduced the idea of microtubule proteins. So that will not be the focus of today's presentation. But also parts of the presentation today. But I hope to drive home this mixture of basic science and then disease and injury. To see that these two arenas of biomedical research are really not at all separate, but are intimately linked with one another. So let me start out with providing you with these incredible cells, neurons, that are arguably the cell type whose polarity, whose exaggerated morphology, across nature of cell type, whose morphology, whose asymmetry is most intimately related to functions they serve. Now, we all are partial to the cell type that we study, but I think I justify an argument that in terms of cytoskeletal challenges, neurons are the most interesting, the most challenged. We really have to come up with the most creative solutions in order to elaborate microtubular ways, other cytoskeletal ways that are sufficient to meet these challenges. And in particular, neurons aren't just polar, they're bipolar. They generate multiple types of elongated processes and axon, multiple dendrites. And these processes are different from one another all the way to discussing this next few days. Axons are long, slender processes, highly specialized to transmit information over long distances. And dendrites are multiple number, thick, tapered, highly specialized to receive the processes of information. And so many of these challenges, the architectural challenges, transport challenges, are fulfilled by microtubules. This is another schematic that illustrates some of the things axons have to do. A couple of things I want to stress is that axons grow, they develop, but they also retract. Now, retraction is not just a pathological event. Retraction is also a highly physiological event. I'll talk more about retraction in a month when we talk about proteins. But when the nervous system develops, the number of axons, axonal branches, really overshoots. Nobody quite knows why. There's more axons than the developing nervous system needs. And a great deal of cooling back of retraction has to occur in order to ultimately result in the fully wired, appropriately wired nervous system. And that structure at the tip, the growth cone that we talked about a bit, that's a high motile structure that forages to the end. It knows where it's going. It has turns to take. The movement of the growth cone is an active base of that. But the turning of the growth cone, that becomes a microtubules. So axon grows microtubules. Axon retraction will talk about growing microtubules. And then growth will turn. A critical part of the talk for this morning is axon ablation. So another thing that happens is that developing axons will grow branches. There's really two types of branching. One is bifurcation of the growth cone, just slits or multiple branches emerge from the tip of the growing axon. But the type of branching I think that's predominant in the case of many sorts of neurons, and I think more interesting, is called the interstitial branching or collateral branching. And that sort of branching occurs along the side of the axon. Let me talk this morning about the mechanisms of that branching. So, with the process of microtubules separate, there are some basic science issues I'll be talking about, branching is one of them, but also the disease situation. As I mentioned, hereditary spastic paraplegia really comes into play, because here's a disease, it's not a terribly common disease, but it's a disease that afflicts thousands of people around the world, and it's a debilitating disease that causes people to lose their ability to walk properly, to motor neuron disease. And I'm also going to talk interestingly about telepathies, because I have a theory, I have a hypothesis about telepathies such as Alzheimer's disease, that involves these separate proteins in a very unexpected sort of way. And this is all about the issue of too little separate or too much separate. Because if you think about it, neurons contain a lot of these separate proteins, a lot of spastic molecules, there's a lot of common molecules. And I think there's around 7 or so total separate proteins. There's common 1, common 2, digit 1, digit 2, in addition to spastic. And a total volume of these separate proteins is enough that in the test tube would completely break the microtubules down to subunits. So that neurons are carrying around dynamo, they're carrying around with them enough separate proteins to destroy microtubular arrays. So that demands a system of regulation. That demands controlling the cell, preventing the available separate proteins from moving otherwise could do. So with that in mind, with all the important functions that cellular proteins perform, I think you can understand there's not enough cellular. The neurons are trouble. But also, there's too much cellular. The neuron could also be potentially more. So here's the microtubule again, this terminology. The microtubule is this polar structure, plus and minus where it is along the length. We'll talk this afternoon more about the molecular proteins that recognize that polarity, and order proteins that organize microtubules relative to that polarity. And remember this movie that I showed a couple of times. This movie generated electrical ground. This is the movie showing transport, very similar to the neurofilament transport that Roy just showed. And we talked about various features of this method. And the feature that's most relevant to the talk this morning is that the microtubules that you see moving in these sorts of wide dimension paradigms are all very short. 5 microns, 6, 7. Certainly, we've never seen a microtubule move that's been longer than 10 microns. Now, if you think about what microtubules do for neurons, particularly for axons, it's really important that they're long, that many of them are long, because microtubules form these long distance railways for mitochondrial transport, which we're not calling that, faculty transport, brain. And also these long microtubules are the compression-bearing structs, the architectural elements that permit axons to take on the long shape. So why is it that only we see very short microtubules when we have axons? Well, probably all microtubules have too much drag, too much friction, and they're immobile. But the movement of microtubules is really, really important because remember we talked about during the initial formation of axons and also dendrites, it's the directionality of that transport that establishes the microtubular polarity patterns. They're so important. What makes the axon an axon? What makes a dendrite a dendrite? But those microtubules are moving, whether plus end leading or they're minus end leading. The other crucial function of this microtubule transport is there's very little any tubular synthesis in axons. The tubule has to come from somewhere. Even if the majority of microtubule cycle, microtubule elongation occurs in the axon itself, that tubule has to come from somewhere. And this ongoing supply of short microtubules supplies that tubule. So the fate of these short microtubules is going to be either one of two things. Either they're going to disassemble at some point to yield their subunits so that the neighbors can get longer, or they're going to get longer. And once they get longer, then they become stationary until a stubborn protein comes along and breaks the long microtubule into short pieces, and now once again, those short pieces can move. So that's the idea. We call the model cut and run. And what I've been interested in over the years, a variety of different proteins. I've studied fundamental proteins, I've studied nucleation proteins. But really the two categories of microtubule proteins that interest me the most are the motor proteins that transport the microtubules, and then these cellular proteins that break them up into short pieces. So when I say cut and run, the cut is the cellular element and the run is the transport. But I think the style occurs all the time throughout the neuron at some low level in order to carefully modulate the number of microtubules that are short, the number that are long, and in so doing, the proportion that are expansive, the proportion that are moving. But I also think there's some key sites in the neuron where microtubular transfer is particularly active and particularly important. One region is here in cell body, particularly in the central zone. So remember, microtubules can be nucleated, obtain their appropriate mass structure, and that's presumably occurring in the central zone of the cell body. And once nucleated, those microtubules have to be released. They have to be cut, severed from the central zone, so that can be mobilized to cytoplasm. So that's one key site. Then also, I think in growth hormones, I'll show you some examples. Remember a couple of days ago, I showed you that stall growth hormone? We had some interesting questions about that huge bundle. Well, when the growth hormone stalls against that huge bundle, when that growth hormone decides to start moving again, that bundle is going to break up. And little short microtubules are going to shoot out of that and into the peripheral regions of the growth. That's a microtubular cell. And then also, that's most importantly, is branch formation. Because the parent axon is populated by this mixture of long and short microtubules scattered throughout the parent axon. And there's just not enough of short microtubules to somehow waylay into that uniform branch, but rather what has to happen is for the microtubules in the parent axon to break up, to create enough microtubules to continue to supply the parent axon, but also supply that uniform branch. So branching becomes a very crucial event that's dependent on microtubule cell. Let's take a look at these several proteins. Mechanistically, they're small. What's known about these several proteins is that they're axons. So 6 of these have to get together. It's a triple A proteins, enzymes that all form axons. So 6 of these have to get together for kind of like stops of axon. And it's always been a little bit of a mystery a couple of years ago how it is that hexamer breaks the microtubule, because the hexamer is really a little too small to fit around the microtubule. The old idea is that maybe it would fit around the microtubule. It's an ATPase, energy transfer down, then it creates torque, and then we break the microtubule. So it's too big to fit on the outside. Maybe it fits on the inside lumen of the microtubule, but then the question becomes, how does it get in there? So recently, fairly recent, a couple of years ago, Antonina wrote a chapter in Raphael's lab, and it worked itself beautifully. And the way that it seems that it works is that this capsule lies really on the outside of the lattice, sits on the lattice. What that capsule is going to do is actually start pulling on the subunit. So in the wall of the microtubule, it starts pulling on the subunit. And then the lattice will start to give and ultimately break because you yanked out one of the tubular subunits. And that's how the microtubule proteins appear to work. So it's ATP dependent, and it's dependent on this axonalization process. Now, if you, yes.
The function of these proteins is completely mechanical, or they do some chemical changes also?
Well, I'm not sure. The question is this a completely chemical thing, or is it a physical thing? And to be honest, I think it's a false dichotomy. I mean, I don't know that there's a difference between chemical and physical. It's a physical process of breaking the microtubule that depends upon the chemical event of ATP hydrolysis. Ultimately, I think what you're going to see, what I'll propose to you anyway, is that through the process of specificity of the various cellular proteins, you're going to create global differences in microtubule with physical and chemical. So we'll get more to that.
Yeah.
Yes.
How many molecules of cation or fraction or whatever do we need to break one microtubule?
Oh, just one. Yeah, that's my understanding, because you really just talk about one supplement, just one supplement in the microtubule brain. So here what we see is we have GFP katana, and what we see here are two different cells. One that you don't see because it's not expressing GFP katana, and then this cell is expressing GFP katana. The unexpressing cell, you see a nice beautiful microtubule brain. And look at this cell. Here's your GFP katana expressing cell. And the microtubules have really broken up in very short pieces. And as a result of this ongoing saccharine microtubules, notice that you really substantially reduce the total microtubule mass. So when the saccharine is allowed to go on, keep breaking, which can ultimately lead to a very depolymerization as well. So the saccharine proteins are very potent, very good. So here are a couple of early experiments. And what we've done here is to look at the role of katana in the release of microtubules from the centers. So katana was the very first microtubule saccharine protein discovered. It was named after the Japanese samurai sword, katana. And way back when, this was 1998, 1999, before we were doing siRNA, before we were doing sort of sophisticated techniques, what we did is we got a function block antibody with block injection. So you have to inject these antibodies with a bunch of neurons. And we used a protocol, a drug protocol, so it's a little bit complicated. The cell body is full of microtubules. You depolymerize the microtubules of one of these drugs. And if you rinse out the drug, you get a burst of microtubules coming right off the center zone. And then you add a little bit of the drug back so that you don't have any further assembly. The little burst of microtubules, it'll eject right around the cell periphery. That's the molecular motor protein moving the microtubules outward. So here are the images you can see. Here are the microtubules gathering around the cell periphery. But if along the way you inject a function blocking antibodies to katana, the microtubules stay in cluster. And notice in the more favorable side, you can see the center zone. It's there on the arrow, and the microtubule system remain on the center zone. So these were our earliest indications that neurons, katana is essential for microtubules to be released from the center zone. Now here are those skull growth plates. So here's the skull growth. These are live cell images. And these are neurons injected with related tubule. And you can see that normally speaking, that moves bottom. And then when the skull growth starts to move again, I hope you can see this lamellar portion. You see all of these short microtubules they're scattered out in this lamellar region. Now look here, this is where we injected the function blocking katana antibody. This lamellar region, I hope you can appreciate this, it's just empty. There are none of these short microtubules whatsoever. So what these two studies show in this function blocking antibody katana is that two different elements of the axon in the growth cone, back in the cell body before the axon, the center zone. Microtubule cellular katana is really essential for releasing the microtubules so they can become mobilized, so they can move into the axon, then out of the growth cones that microtubules can break off that root bone become mobilized so that the axons start growing again. Now, my favorite idea, remember I told you the other day that I had this notion about branching of axons. My idea about branching of axons was that in order to get into branch, there has to be a lot of microtubular separate to be able to break up the microtubular parent axon and mobilize the microtubules. So my favorite idea for one would be if I overexpress GFP time in these cells, I should see a lot more branch. That was my hope, and it didn't happen. So we have this axon here, we overexpress the common, and we certainly break up microtubules, your axons are a little funny, but we did not create more branches. So that was a bit disappointing. Yeah. Is the length of the axon the same? No, if you express high levels of common and you break up microtubules, oftentimes that can be a lot shorter, or you wouldn't have an axon at all.
Yes, so the question is that if you temporarily activating having an axon, non-axonal reform, do you induce branching?
That would be a much better experiment to do. If you could somehow temporarily, spatially activate the time, and we haven't been able to do that. Let me show you a few more experiments. So, what we came up with was this idea. I'll give you the data in just a moment. The idea, I actually presented this to you a couple of days ago, is that what's happening here is that it's not just a matter of having a lot of axons. I mean, it's already a lot of axons, but the idea here really is regulation. And how you regulate our hypothesis is that the common is very sensitive to the levels of power in the microtubules. But there's a lot of power in the microtubules, and then the common is really not so good at separating at all. So what has to happen is that the tau has to become phosphorylated so that it's released from the microtubules and not the common inside of the microtubules. So I would posit to you that perhaps mechanistically what's happening is that the signaling event that's giving rise to a new branch, what that signaling event is doing is it's phosphorylating tau. So that tau is released from the microtubules, and now the time you move inside of the microtubules. So let me show you a little bit of data that can support that idea. These are the sorts of experiments that we're going to get about from other studies, which we take simple fibroblasts and we use them as a workbench. So what we're going to do here is we're going to express various forms of tau. This is 4BB for this. This is tau without microtubular binding domain. This is another tau without microtubular binding domain here. But this is just microtubular binding domain. This is tau without microtubular binding domain. And tau is very, very protective. So if you express the tau with the time, you really don't see any microtubule separating at all. If you express tau without the microtubular binding domain, you see a lot of separating. So the tau has to be bound to microtubules in order to protect it against tau. So the presence of tau on the microtubules is protective. We've actually done the same experiment with various methods. If you do it with MAP1B, for example, you don't see any protection. If you have to do it with the tau parameter, MAP2 protects, MAP4 protects. These are very similar structures to tau. So if we do the inverse experiment, now what we're going to do is we're going to take neurons and we're going to use siRNA to deplete MAP1B, and then we're going to overexpress the common. Notice in the control, if you express the time of just 12 hours, notice how the cell body reacts to the microprocesses. They're much more efficient in microtubules. This is a microtubule study, much dearer. Whereas the axon remains pretty bright. And the reason why the axon remains pretty bright is because it's got a lot of power attacking the microtubules. Notice if you deplete MAP1B, the axon's still pretty bright. Depleting the MAP1B hasn't really made any change. But if you deplete tau, not only the axon becomes vanishing bright in the microtubules. Because now when we overexpress katana, the microtubules have no resistance and no protection. This is an immunostate. This is a quantitative immunostate. And what we've done here is this is a ratio that shows dephosphorylated tau to total tau. Dephosphorylated tau, remember, is going to bind the microtubules, and phospho-tau is going to come off the microtubules. So notice that this region here in black, that region represents an area where the tau is particularly phosphorylated, particularly unbound to the microtubules. That's right. That's exciting having branch formation. So another piece of evidence that supports the idea that tau phosphorylation is regulating access to microtubules. Let's not get so focused on that we forget that there are other cell proteins as well. And this is where hereditary spastic paraplegia comes in. About 40 to 60% of all the cases of hereditary spastic paraplegia have long been known to be caused by mutations to the SPG4 gene, which encodes a protein called spastin. This is usually an adult onset disease. The average age of onset is, I think, about 42. Usually, people younger than 20, 21 are not going to see typical hereditary spastic paraplegia. There's spasticity in the lower limbs, problems walking, and degeneration is really quite specific to the corticospinal tracts. So mutations to spastin are causing degeneration, specifically in the adult corticospinal tracts. Very interesting about spastin. If you look at the spastin molecule, here's the AAA feature. It's very similar to tau. And that's how the label barley generally around year 2000 or so took a look at the spastin molecule. And she said, that looks an awful lot like tau. And sure enough, she made a construct, she extracts it in cells, and it breaks up the mitochondrial. Even better. It's a very, very potent mitochondrial cell protein. What's curious about spastin is that there are two major isoforms. These isoforms are produced by two separate organisms. So the longer isoform we call M1, that's encoded by the first separate organism. And then M85 is just a little bit shorter, and that's encoded by the second separate. Now, the vast majority of spastin anywhere in the body is a slightly shorter isoform. So it's the M85 or M87 crystals. We call it M85 mouse, rats, and then in humans themselves. Good fibroblasts today, we express the time for about 12 hours and fasting for 4 months' time. Here's the controls of our Bombay tubules. And notice there's a bit of a difference in the cellular pattern. With Huntington, you see a lot of the short microtubules, but you also see a lot of long microtubules. And look at fasting. It's all short microtubules. So the properties of these cellular proteins are identical. Expressed on the identical cell type, and the result of microtubule patterns a bit different. Now, remember I said that I need experiments and overexpress the content, and I was going to get a lot more branches. My dream was crushed. I can see more branches as well. Along comes fasting the lesson, because if I overexpress fasting, look at all these branches. This is the GFP fasting, and here's the tubular state. And normally speaking, here's some control axons. Well, kind of little branches along the way, these axons are relatively short. But look at these fasting overexpressors. Lots and lots and lots of branches. If we knock down this fasting by siRNA, we severely reduce the number of branches. Overexpress fasting and severely increase the number of branches. So whereas Huntington probably has a more complicated pathway to go through to create a branch. Spasm here have a very simple pathway. There's no spasm available in the branches. If you elucidate for the common spasm, common has a relatively smooth distribution. It just appears to be equally distributed everywhere. Whereas spasm, notice that you have concentrations. You have concentrations at tips of processes, and then also here at these branch sites, there's another branch site and a concentration of spasm. So, again, two different modes of branch. Katana remains equally distributed. I think it's that focal dephosphorylation tau that says to the axon, branch there, don't branch there. Whereas the spasm appears to be an accumulation of spasm at specific sites that yields branches at those sites. Here we see some branch sites that don't seem to have spasm. I'd say one of the things is going on with those branch sites. One is that the branch is already formed, so now the spasm is no longer required there. But alternatively, those branch sites are common branch sites. I didn't have to have spasm accumulation. Yes.
This spasm organism due to the cellular microtubule, but there is also branching of the axon. So we only the entry of microtubule into the branch has to be decided by a lot of other factors. What is surrounding and active growth for not something, or is just entry of microtubule to decide that the branch.
The question is a very good one. Is branch formation exclusively caused by microtubule cellular body cell proteins? No, of course not. Branch formation is a complex process that requires actin-based mechanisms, membrane-based mechanisms, a coordination of cytoskeletal events. All of these work together in order to get a branch. So I don't need to apply simply, only, exclusively microtubule cellular. And that might be another contributing reason why simply overexpressing the common, perhaps it's too big of a dream, too big of a goal, to think that simply overexpressing the time would make branches, because it's not just the cellular microtubules, it's a combination of many, many different events. So, the fact that overexpressing spastin does cause branches, I think would tell us that whatever other machinery is required to make a branch in these cultured neurons is fully available and fully prepared to do so. It's not our
So, how overexpression of spastin, which causes more severe microtubules, can maintain longer axons?
Say it one more time.
So, in case of overexpression of spastin, we have more severe microtubules. So, how they are maintaining the length of the axon, because they are longer than the right definition, even if they have no overexpression.
Well, with everything with overexpression experiment, it all depends on the levels. The question is, if you're breaking up all the microtubules into short pieces, how do you maintain the axon? Well, if we push this harder, if we broke up all the microtubules into short pieces, you could not maintain the axon, because the long microtubules are in compression near the struts. The long microtubules are the substrates for organelle transport. So, an overexpression experiment has to be done with a very open mind to these sorts of things. So what you need to do is you need to achieve the appropriate level of overexpressed proteins to create sufficient short microtubules in order to generate branches without destroying or altering microtubular in such a way as to somehow curtail the other functions of microtubular was performed.
There's actually a branching is being made when there's a need for it to be a means to work. So, okay, fine. When start being regulated by Charcot and pick up advantage of the sporadic. There has to be a regulatory mechanism for spasm as well, right? Because if they don't just form branches in this organized, then it has to be regulated. So is it known, the regulatory mechanism for spasm?
The regulatory mechanism for spasm is really the next frontier. Maybe we'll talk about that a little bit more as we move forward toward the end, but very little is known about it. Just recently, a phosphorylation site was discovered at the time that may be important for regulating it. With spasm, that same phosphorylation site is not present. To the best of my knowledge, there is really nothing known about phosphorylation sites or other sites, or sometimes certain molecules will fold on themselves in order to auto-regulate their function. To the best of my knowledge, nothing like that is known about spasm. You have to have hexamerization. That's one level of regulation. That these individual spasm molecules are simply floating around unhexamerized form, and obviously they become microtubules. The way they become hexamerized is they use the microtubule itself as a workbench, so that they sit on the microtubule, and the presence of the microtubule enables the supplements to come together and form the hexamer. So that is one mode of regulation. We're talking a little bit more later about the post-translational modifications of the microtubule itself, not regulating the spasm or the time, but making the microtubule more or less sensitive to the time of the spasm. So, we have a few hints. We have the tau protection for time. We have the acetylation, the deteroxidation status of the microtubule. But much more needs to be done about regulation and processing. Not really on its own.
So what happened if we expose all of these reverse aspects? So do they exactly manage to really regulate the type of technique?
It doesn't doesn't seem to be the case that common spasm interact in known ways. We used to wonder a little bit, given that they're similar, could they say co-axons? I don't think they co-axons because their distributions are so different. And we used to wonder, for example, with our function blocking antibody, because the phenotype is so strong, might that antibody also interact in addition to the spasm, again, to the best of our knowledge, nothing is known about common spasm regulating one, except maybe one thing. But if you knock out one, say you made a genetic animal, not one of the model, use one of these SNR procedures to knock out common spasm. I think allows the others to come up to confidence.
So I have two questions. So, is there any difference in the localization of the two spasms? It is like when you do the isomers of the spasm, they have LLS, one isomer, LLS, LLS, another one is also one.
I will talk about two isoforms in a bit. You cannot discern the two isoforms with separate antibodies as of yet. We're trying to develop separate antibodies, but it's tough. It's a tough challenge. I think the properties of the two isoforms may lead to different distributions. One of the isoforms, the full-length isoform, for example, has a very highly hydrophobic domain, such that you might think, going back to Dr. Roy's talk, you might think that one isoform, the one isoforms contain axonal transport, whereas the other isoform is transporting slow component B axonal transport, which might lead to very different distributions. But also keep in mind that the longer isoform is really only expressed at any detectable levels under very certain circumstances. I'm going to talk about that very shortly. But the vast majority of spasm is just a shorter isoform.
So, another thing is that in case of overexpression of spasm, can we speculate that because the microtubules are now short, there is redistribution of proteins to regions as a result microtubule and the protein that it is.
You know, it's very important. If you think about the manifestations of the microtubules being short, and maybe the redistribution of proteins. Now, it's not the redistribution of maybe one of the most important proteins. You're going to hear a lot more about the so-called plus proteins tomorrow. You've got a lot more plus ends. That means you've got a lot more plus steps potentially. And the plus steps really have the property to interact with cortical structures. If you have EB1, EB3, they can interact with other plus steps, the clips, the clasps, they can get captured. Another one of these two presentations is all on the role of the plus steps in the growth. So I suspect that maybe what's happening in branch formation is not just the mobilization of the short microtubules. It's the fact that there's some more plus elements and the accumulation of all these plus steps, and then those sorts of things are essential. So these are wonderful questions. Remember, nothing in biology happens in isolation. You don't just knock out a protein, overexpress a protein, or break up microtubules. No, only one thing happens. Well, now you're short enough to move on. Sure, there are a lot of other things that are happening as well. It's a very good.
Does branching triggers triggers branching?
Does branching trigger severing or severing trigger branching? I would say severing triggers branching, sure. But I wouldn't dismiss the possibility that branches could induce settling as well. I mean, say, for example, you start to form a branch and get that actin-based structure pulling and pulling. You used to see a microtubular literature called bending and breaking. That what people would study was maybe only or Carolina stores, and these both microtubular people would look at the leading active cells and get the impression that because the active structure is pulling on the microtubules, they get phenomenon called bending and breaking. Well, in neurons, microtubules down all the time, they don't break. I don't think microtubules break because of that. But maybe that by virtue of that, you enhance the access to the cellular proteins. So I think the potential will go both ways. I like that question. Now, what really causes branches to happen? What causes branches to occur are these complex pathways. And there are growth factors that induce branches to form. And one of them is called basic FGF. FGF is body mass growth factor. And it's a very potent factor which will cause a lot of branches to form. And I know that your eyes may not be able to appreciate all the details here, but this is a lot of Western blot that we get. And to make a long story short, if you take cultured hippocampal neurons and treat them with FGF, you get a lot of branching. Mechanistically speaking, what's happening? Well, you get an elevation in putamen levels, you get an elevation in spasm levels, and you get lower autonomous storage. So really, at 3 different levels, who are wondering, well, maybe FGF causes branching through a spasm mechanism, or maybe FGF causes branches through a putamen mechanism. Maybe it's the tau pathway. As it turns out, this particular growth factor really causes changes consistent with branching in all 3 of these. There are a lot of factors that cause branching. There may be, naturals or different things. And I think if you survey these different factors, you may find certain factors work through a spasm mode, and other factors work through a putamen mode. But at least at present, it appears to be that this factor works through both. So here's the slide I showed briefly a couple of days ago. There are 2 modes of branching. Spasm mode, which appears to be an accumulation of spasm at particular sites that give rise to branches, and the putamen mode that works through that tau phosphorylation that we discussed. Yes, that's your question. No. Okay. So what else besides tau might regulate these cellular proteins? And one of this is this tubulin cone that we brought up the other day. So maybe certain of these cellular proteins have preference for disseminated microtubules, heterocyclic microtubules, polycontaminated microtubules. And remember we showed that forms. Now these are the tyrosynated and detyrosynated domains. Acetylation looks a little bit different. The acetylated tubulin seems to appear in the form of these patches along the length of microtubules. So nowadays there are some tools for manipulating this acetylation pretty effectively. So HDAC6 is a deacetylase, and we can overexpress HDAC6. And then tubicin is a drug that inhibits HDAC6. So if you overexpress HDAC6, you're going to get a lot less acetylation. So this is an elucidate for cellular tubulin. This guy right here is overexpressing HDAC6. Notice that it contains almost no acetylate tubulins. It contains a little bit. Then if you treat the tubicin to inhibit HDX6, then you do your cellular tubular state, they're screaming bright. So we have experimental ability to increase acetylation or decrease it. And to make a long story short, if we elevate microtubule acetylation, then we get more acetylation. If we lower microtubule acetylation, we get less acetylation. And if we look very specifically at where acetylation occurs, this is ketone, this is fasting. We look very specifically at where acetylation occurs. Notice with ketone, this is acetylated tubular. I hope you can see this. It's real hard to see. But the acetylation occurs where is acetylated tubular. Whereas this fasting seems more random. So here's up, and in this region you don't really see the acetylated tubular state. So what the conclusion here is that ketone really seems quite specific for acetylated tubular. Microtubules are rich in acetylated tubular, whereas fasting doesn't really seem to care so much. So we take these studies in neurons. Yes.
So that would mean that catenin would lead to more stabilized branching because one would be more stable forming the branches.
Yeah, that's a really good point. Because what is the purpose of microtubule cell? And at least with regard to the cut and run, the purpose is to create short, mobile microtubules that can move into a new branch, that can move out from the centrosome, that can live in order to carry out a purpose. So if you imagine breaking up a highly dynamic microtubule, it's just going to disassemble. So there's not much reason for doing so. So the idea is, if you can target the microtubule cell to more stable microtubules, then yes, those short pieces will persist. They will live in order to be able to transport into new branches, into axons or dendrites. So having this mechanism for preferring stable microtubules through a preference for acetylated tubule makes good sense for that reason.
And it could be like catenin gives rise to acetylated microtubules as such, and they could act as mutating centers using the fibrillary monomers generated by
Yes, exactly. By ketone having a preference to break up acetylated microtubules, the end result would be a high number of short fragments that are stable enough to resist depolymerization and live another day to active nucleation sites. Exactly.
Does cell have different affinities for acetylated does cell have different affinities for acetylated and non-acetylated?
I don't know. It's a good question. Does tau have a different affinity for acetylated or deacetylated tubulin? I don't think there's any evidence on that I know about. So moving on to neurons, we did our two experiments. Remember, with HDAC6, we can substantially lower the content of the cellular tubule. Then with tubicin, we can significantly increase. This is 24 hours of ketone overexpression. Now look here. This is HDAC6. We've overexpressed ketone for 24 hours. And notice the microtubule state is still very robust. The microtubules are really not being acetylated very much because they're not acetylated. By contrast, you barely see this state. In the tubicin case, because in the tubicin case, you have made the microtubules very, very acetylated. way. You made the microtubule, and that happens backwards. So in HDX6 case, you made the microtubules very unsettled. So yeah, and so the difference is you've got tau crest. The difference is that with the tau crest, for axons, it doesn't care so much, it's only the dendrites that care. So it's a little bit of a complicated situation. So that makes the story a bit complicated, but for axons, notice that all these bars are similar because whether you have the control, the HDX6, the tubicin, the common, whatever you do, it's very similar because the tau rich, whereas dendrites really follow the same rules as the fibroblasts. So the control, the microtubule levels are high, the HDX6, the levels remain high. Tubicin, now with tubicin, we made the microtubules very separated. Now here, with ketamine, we lowered the levels. With the ketamine plus the HDX6, now the levels do not diminish because the microtubules are very unsettled. But with the tubicin, now we get decrease. Now we get a decrease. If you overexpress tau, since the tau was the axon and dendrites, so it's very protective, it doesn't matter what you do. So the neurons are a rather unusual situation because they have to pay attention to different factors. They have to pay attention to acetylation status and the presence of axons and tau on the tubules. So various levels of regulation. One is the presence of various separate proteins, about 7 total, with some different properties. Another is the levels and distribution of these separate proteins. Some are widely distributed, others may be concentrated at branch points, for example. The sensitivity to tau and other sorts of factors. And again, it's not very exhaustive. Spasms, for example, may be very sensitive to the presence of, say, for example, double cord microtubules. So this is another level of regulation that needs a lot more work. And then acetylation, other tubular modifications may be a factor. Carcinogenically, since really excellent work showing that spasm is highly sensitive to the levels of how tubular microtubules, for example. And then another possibility is the regulatory degradation of these proteins. Now, what about disease? I want to talk a bit about hereditary spastic paraplegia and a bit about tauopathy. And I'm going to ponder, is hereditary spastic paraplegia not enough suffering? Is that really at the heart of it? And is tauopathy really too much suffering? So, getting back to hereditary spastic paraplegia, this is autosomal dominant disease, mutations in the spastic gene, very specifically causes degeneration of orbital spinal canals. There are no developmental problems, no problems with performance. So it's highly specific to the central nervous system, it's adult onset, caused by mutations to spastic. Now, here we have a mystery. Why is it that this is adult onset? Why is it only the cortical spinal canals? Now, the community studying hereditary spastic paraplegia, mainly are people who come after the disease, not from a microtubular background. And this is really a good lesson about how having a background in police science and coming at these issues with a large body of basic science information can really be helpful toward having solid insight into the etiology of patients. So, the theory that most people in the community really latched on was a loss of function scenario, a haploinsufficiency. So how many know here know haploinsufficiencies? So what haploinsufficiency? Can you explain what's haploinsufficiency?
One dose of the same gene is not sufficient.
Exactly. So you've got two spastic genes, and the idea is one of them is mutated such that the protein created by that gene, that mutated protein, doesn't work. Then you're at the mercy of roughly half the amount of total protein, and that's not enough. That's not enough to keep you healthy. And if you look at the hereditary spastic paraplegia literature, ever since spastic was discovered to be a settling protein, people have latched on to this haploinsufficiency idea. In truth, not all that many diseases are due to haploinsufficiency. It's actually kind of rare, particularly for enzymes, because usually enzymes are made in excess, so that you presumably have enough of it, even if you only have half. But this becomes even more mysterious for spastic, because spastic would be presumably especially important for development, right? There are a lot of things that have to happen during development, branch formation, and growth hormone motility, release from the cell chromosomal microtubules, a lot of different developmental events. And yet, developmentally, there are no symptoms. The patients are perfectly okay. Moreover, why would only the corticospinal tracts be sensitive? Why not, say, the sciatic nerve, which is a peripheral nerve, but is quite long? I mean, some people make the argument that, the reason why it's the corticospinal tracts is they're really quite long. The long motor axons. They go to the limbs. Well, there's some long peripheral nerve axons as well. So my feeling is that most people in the community who did not really have a basic science background, once they saw, it's a microtubule settling protein, there must be something wrong with the microtubules. There must not be enough microtubule settling. So what we did is we looked at a few parameters in this. One is we looked at the absolute levels. So this isn't just portions. Absolute levels. We took our antibodies and we calibrated them against the common protein. And during development, there's katana, there's spasm. And the levels during development of katana progressively go down. The levels of spasm are pretty steady. The levels of spasm are not going down appreciably. So one argument you can make is that all the way during development, maybe katana can compensate for spasm. But then once you reach the adult years, then maybe not. Maybe there's not enough spasm and katana in combination to induce sufficient cell to keep the axon healthy. So maybe. But what I'd like to propose to you is a gain of function. Now, let me tell you, if there's any person anywhere in the world who should be emotionally invested in a loss of function mechanism for hereditary spastic paraplegia, it should be me. Because I put so much of my career into the importance of microtubule settling in the nervous system. And yet, I just don't think it makes a lot of sense for the reasons I just explained. Let me tell you why I think a gain of function makes sense. What do I mean by gain of function? Anyone know what gain of function is?
This is functionally more active.
Not really. Acquires a new function. So when the spastin mutant, when the spastin protein has the mutation, it acquires a new function. That sounds like a good thing, but no. When I say it acquires a new function, I mean a bad thing. I mean it's doing something it's not supposed to do. So loss of function diseases are when there's not enough of the protein to carry out what it's supposed to do. A gain of function disease is when that mutant protein is doing things that are bad, that it's not supposed to do. And the key to all of this mystery, I think, is all about these two isoforms. So remember the shorter isomorph that comes from encoding by the second coder, almost all the spastin that you've ever found in your body, and it's not just in organs, it's throughout the body. It's in skin cells, it's in all organs. The vast majority of that spastin is the shorter isoform, M85, Neurodegeneration, M87. This longer isoform, when we Western blood, this is cerebral cortex, this is spinal cord. This is E18, P0, embryonic 18, that's newborn, that's P12, this is mouse, spinal cord. Here's M85. M1 will appear up here. You just don't see. You just don't see. Now look at spinal cord. Spinal cord is the cord of spinal cord. E18, nothing. P0, nothing. P12, nothing. Look at that. You see that? In the adult, you start to see M1. Adult spinal cord, adult cortical spinal tracts. My theory is that when M85 bears the mutation, it may not work. It may not sever microtubules, but it's not causing any problem. When M1 bears the mutation, now you've got a cytotoxic protein. Now it can cause problems. Because that is present in the right place at the right time. Adult spinal cord. And here you see that. That's even better. You see that one. Let's take a break for 10 minutes and then I'll finish up.
Yeah, please
Take a break for 10 minutes. And the difference between M1 and M85 is a region that's really very hydrophobic, and a region that might be very prone to misfolding. And as you remember from previous talks from Dr. Brady and others, that when proteins misfold, then they can start imposing some real toxic effects, at least potential. So what we've done here is we've made some mutant forms, and these are just stock forms. We've just simply taken the M85 and M1, and we've made what is amounts to a truncation mutation because of a whole variety, actually about 200 or more different mutations in spasm that give rise to the hereditary spastic variation. Some are folate mutations where the patient has the entire spasm molecule, which is one mutation, and others are called truncating mutations that result in shorter proteins. So we created some of these shorter proteins. We did a couple of experiments. And this experiment you see in the lower panel was done with Brian O'Keefe and Scott Brady. Very similar with axoplasm studies that Scott showed a few days ago. And what you can appreciate from this is that with the M85, really no effect. So we've used the split axoplasm with the mutant form of M85 in both forward and backward transport acetylcholine appears pretty normal. But here, notice the downward slope in both the anterior gradient and retrograde transport. So, this mutant form is faster when it uses split axoplasm. It seems to be creating a deficit in fast axonal transport. So, for haploinsufficiency people, this is a very strange sort of result. But gain of function makes good sense. Very similar to what Scott Brady reported, say, with mutants with tau fibrils, with mutant proteins, studying Huntington. Here, it's cell biological experiment, where we took control neurons. Here, you see the express the M85 mutant. Looks pretty good. Long axon, some little toponia, processes, not much difference in axon length. But look when we express the M1 stop. The cell body is smaller, the axon is shorter, and it just doesn't look good at all. Now, this was mutant mouse M85. We reported this a couple of years ago. More recently, in my lab, what we've done is we've generated a whole variety of mutations using M87, specifically for human. And when we've done this, we've made mutations. We've made the exact mutation. We never just created this artificial stop protein. But we've put the exact mutations in a whole variety of different patients in these constructs. And there are even a few mutations that are actually not function blocking. They're actually not function blocking, but they still cause the disease. And that within itself should obliterate the haploinsufficiency, the loss of function mode. Because those mutations do function properly. They're not in the ATP region, they're in some other region of this vascular molecule. Yet they still cause the disease. And when we work with a human, and we introduce these exact mutations, these mutant spasms are far more toxic than you even see here. We can barely express them when they kill cells. We express them intracellular, and the flies just look terrible. You've got to use specific promoters that induce the expression only the adult fly, only in certain neurons, or you don't even have a fly. So I think what this shows is that spastin is a molecule, at least M1 spastin is a molecule, that if it's misfolded, it's tragically toxic, which is probably why evolutionarily spastin developed that second start codon, is so that in the vast majority of cases, the cells can dodge, even playing fire with M1, because M1 spastin is a protein that is so prone to disorder. Now that begs the question that I'm sure many of you are thinking, why is it that adult corticospinal neurons will start expressing M1 spasm? Why do they make this trouble for themselves? I don't know. I don't know. Does it have some kind of function that evolution needed to hold on to? That the corticospinal neurons need to have some of this M1 spasm? Or in some way it was just a mistake, just a tragic mistake that was made along the way, that the human body would be far better off and never preserve that initial start code. We don't know the answer to that. But I think, at least in my mind, that these data are really compelling. The haploinsufficiency not only is illogical for this particular disease, but that the weight of evidence is really against it. There's also a fast and knockout models that seems perfectly fine. Knockout spasms in individual neurons and culture, they do okay. Maybe not quite as many branches, but they seem to do okay. All the other neurons of the body seem fine. Patients with hereditary spastic paraplegia. So the idea that there's something somehow derived in the nervous system, when it's also expressing 6 unreceptive proteins, I think stresses credulity. I think this gain of function model not only fits logic, it fits the evidence better, it also puts hereditary spastic paraplegia more squarely in the category of other neurodegenerative diseases, such as Alzheimer's disease, such as ALS, Parkinson's, Huntington's, other diseases that are caused by misfolded proteins. And I bring up this story about haploinsufficiency not to disparage the investigators who favor this model, but I think really to point out the incredible value of basic science. Now, we all contribute at different levels, and clinician scientists who are focused mainly on disease, they have a great deal to offer as well. But having this body of basic science knowledge is so incredibly important, so useful, that when I think back to, remember I said 1993, when I was thinking to myself, how do branches form? How do microtubules get to a branch without calling back to the centrosome? How do they do that? And I thought, well, maybe it's because microtubules are broken up into little pieces. Nobody had ever discovered even autonomy yet at the time. But it made good sense. And at the time that I thought about that, at the time that we did all this tedious serial reconstructions, electron micrographs, thin sectioning, put all those back together again to show that indeed microtubules are separate at branch roots. Was I thinking that someday that would help us to understand hereditary spastic paraplegia? The thought never occurred to me. Never occurred to me. It was really the pursuit of basic science questions, the curiosity, and the wonderful curiosity that I see in all of you, and all the questions that you keep asking us. And so often we say, I don't know the answer. It's not been done yet. It's that curiosity that's going to lead to treatments and cures for all sorts of diseases and maladies and injuries that afflict the human population. So I think that's the broad lesson of this sort of thing. Now, what about too many drugs? Finish up real quick. Okay, fine. So, the idea about tauopathy, let me make this really quick. This is what you see all the time. Tau comes off the microtubules, they become destabilized and they break up. Well, if the microtubules break up in Alzheimer's disease and other tauopathies, it would be terribly tragic. And they do break up. I mean, there's a loss of microtubule mass. And I'm going to tell you, as I've argued, and as Scott Brady has argued, there's really no evidence that microtubules truly destabilize when they lose their tau. There's just nothing. I mean, people have knocked out tau, microtubules don't become destabilized. But neurons contain double cortin, MAP2, MAP1B, MAP1A, stop. They contain a whole array of stabilizing proteins. So here's my idea. My idea is directly launched, directly based on the tau information that I gave you earlier, that if you deplete tau, the microtubules become very sensitive to ketamine. So my theory is that when the microtubules lose their tau, they're becoming progressively more sensitive to these several proteins. So they're falling apart, yeah, but not by being destabilized, by being over-severed. I'm not going to show you all these data, but that's the idea. That's the idea. So here, notice, you've got ketamine overexpression, you've knocked out tau, and you really lost microtubules. So one other thing I'll show you really quickly is we heard from Jim some beautiful data on axon regeneration. Is it conceivable that we can exploit microtubule cell to augment axon regeneration? And what we found is that if we knock out fidgetin, we get a lot more axon growth. A lot more axon growth. These are adult DNA genes, so this is a regeneration system. Fidgetin is another microtubule cell protein. And we get more crossing onto inhibitory substrates. Here's what I think is going on. This is just a hypothesis, and you are the first people to hear it. It's based on a little bit of data you see here. But I think what happens when you knock down fidgetin is you get less acetylated tubulin and a little uptick in total tubulin. I think what's going on is that just as ketamine targets acetylated microtubules, fidgetin targets unacetylated microtubules. Now, why should it do that? How would that work? And my idea is, you've got an axon that's full of microtubules, acetylated microtubules, deacetylated microtubules. Imagine this. The fidgetin is constantly severing the unacetylated microtubules. And that's what enables neurons, as they develop, to accumulate acetylated microtubules, because you're constantly pruning back on the unacetylated microtubules. And that's important for reaching the adult state of having a predominantly acetylated microtubule array. Think of it like a bonsai tree. You know what a bonsai tree is? You're constantly trimming it. By constantly trimming it, very specifically, you are shaping it. And here in neurons, there's ketamine. Ketamine like 1, ketamine like 2, fidgetin, spastic, fidgetin like 1, fidgetin like 2. And they're all working a little differently. Imagine different scissors trimming away at different categories of microtubules. Tau-rich microtubules, tau-deficient microtubules, acetylated, polyglutamate, unacetylated. And that's how you can craft the microtubule array to be what it needs to be in an axon, in a dendrite, in a developing axon, and in an adult axon. What you want to do with the adult axon, when it's injured, is restore it to a more developmental state so it grows. And that's why I think inhibiting fidgetin works to augment regeneration. All that is just a theory. Everything I just said, I'm not going to go through these conclusions, all that is just a theory. I like it. But it's going to require a lot of work to do. So the field of microtubule saprophytic proteins is wide open. These are really, really, really interesting proteins. And if you look at PubMed, you go to fidgetin or fidgetin-like 2, maybe 2 papers published. Almost nothing. You try to get antibodies, they're not available. So these are wide open. Lots known about tau, lots known about kinesin 1. Almost nothing is known about fidgetin-like 2. There was a paper in Nature that was published a couple of months ago on autism, in which the most commonly mutated genes of autism were categorized. Number 3 on the list was Axons-like 2. Huge list, hundreds of genes. The third most common mutation. Now that's a correlation, it's not a cause. But in autism, there are flaws in neuronal migration. And I'm thinking, what if Axons-like 2 has a really important role in neuronal migration? So let's start studying neuronal migration. So I think this goes to illustrate that the basic science behind these proteins, behind many proteins, broadly open. And if you choose to take up the call and to study some of these, the rewards will not only be opening the door to basic science, but truly, that is my way of thinking. That really is the only way to one day solve the diseases and to develop strategies for treating the injuries that afflict the human population. So that said, these are the people in my lab who conducted this work, and thank you for allowing me to go over time. I apologize.
I'm sorry, I have to cut down the questions now, because otherwise there is a lot of mismatch. So we go straight to the holodeck after this, and we come back and have your questions post class when Peter comes back. Thanks, Peter.
Is these microtubule clarity patterns within axons and dendrites that I told you really how fundamental